In this final section of our chapter on specific immunity, we'll be considering vaccinations, different types of vaccinations, how they're prepared, and then some of the suggested schedules for vaccination. Before we get into that, we need to think about the 4 specific descriptors of immune state. Immunity is either characterized as active or passive, or natural or artificial. This really leads to 4 different categories of immunity. Of course, each of these are acquired immunities, so they're specific immunity. Natural immunity is immunity that's acquired through the normal life experiences, and it's not induced through medicinal means. Whereas artificial immunity is immunity that's produced purposely through medical procedures, like immunization. So natural immunity can be either active or passive. Active is a result of a person developing his own immune response to a microbe, whereas passive is as a consequence of one person receiving preformed immunity that's made by another person. The best example of this is immunity that's passed to a baby from its mother. When we consider artificial immunity, we can also have active or passive. Active artificial immunity is a consequence of a person developing his own immune response to a microbe that has been introduced by medical procedures, like a vaccination. And passive immunity is the consequence of one person receiving preformed immunity that's made by another person. Both of these are really the focus of immune treatments. When we receive passive artificial immunity, we're receiving antibodies from someone or some animal. They have short-term effectiveness because the antibodies have a limited period of function. Once the treatment is received, the recipient's body breaks them down and disposes of them. So they just have a limited period of function. Whereas artificial active immunity, that's the vaccination, produces a longer-term effect in general. Vaccination comes from the word for cow in Latin, which is vacca. That's because the first vaccine that was introduced for active immunization was for smallpox, and it comes from cowpox viruses. But since then, vaccines have been developed for many other animals, as well as being produced by bacteria. When we consider artificial passive immunity, we're thinking more about immunotherapy. So artificial active immunity is vaccinations, whereas immunotherapy is where we have preparations of specific antibodies, like preformed antibodies, from another individual or animal, and an individual is treated with these preformed antibodies. Different than vaccination, where our own bodies are creating antibodies to an antigen that we're injected with or consume in some other means. Great examples for artificial passive immunity are when people have an immune system that cannot respond to an antigen, or things like antivenoms that are used for snake bites. So the object of artificial active immunity or vaccination is to obtain the same immune response as we see in natural immunity, using a modified version of the microbe or its components, but also meeting the requirements in this table. So in general, we need to select the correct antigen. It needs to be effective. There needs to be a certain degree of ease for administration. It certainly needs to be safe and definitely cost-effective. Otherwise, we can't get most of the population vaccinated. Table 15.4 gives a checklist of requirements for effective vaccination. The vaccine needs to have a low level of adverse side effects or toxicity so it doesn't cause serious harm. It should protect against exposure to natural or wild forms of the pathogen, its primary purpose. And it should stimulate both antibody B cell response and cell-mediated T cell response. It needs to have a long-term lasting effect, that is, it must produce memory. It should not require numerous doses or boosters, although occasionally we'll see that vaccinations do require this. And it should be inexpensive, having a relatively long shelf life and be easy to administer. Otherwise, again, it's going to be difficult to get people vaccinated. Vaccines can be prepared from either whole cells or viruses, or parts of cells or viruses, or antigenic molecules or subunits from bacterial cells or viruses. In the whole cell virus preparations, we can either have live attenuated cells or viruses. Attenuation means loss in intensity. Or we can have killed cells or inactivated viruses. In the first example here, we have the killed cell inactivated virus route. Heat or chemicals are used to kill that virus. Here we have a bacteria that's dead, and because viruses are not ever really living, we call them inactivated. But they still have antigenicity because they have antigen labels on their surfaces. When we administer these to an individual, the vaccine stimulates immunity, but the pathogen cannot multiply. In the other route, where we're using attenuated cells or less effective cells, you have a bacteria or virus shown here, where the virulence is eliminated or reduced by a number of different means. So we have a virus or bacteria that's alive with the same antigenicity. We administer it to our subject, and then the microbes are actually able to multiply. This allows for a much more pronounced immune response. Either way, both of these whole microbes will stimulate immunity and ideally cause no disease. The other way that viruses can be generated is by using subunits that are derived from bacterial cells or viruses. These subunits come from cultured cells or viruses, or they could be chemically synthesized to mimic the natural molecules. The subunits can be manufactured by genetic engineering. And any of these subunits can be conjugated with proteins from other microbes in order to make them more immunogenic. We call these conjugated vaccines. So anytime we're taking a subunit from a microbe and then conjugating them with proteins of another microbe, then it's a conjugated vaccine. In this figure, we can see viruses, and these subunits here are used to generate a vaccine. In bacteria, there are more components to work with, but we can take any of these different components of the bacteria and we can elicit an immune response. Basically, we're taking the antigenic portion or the epitope regions of proteins or other molecules and injecting just those. In this case, the antigen stimulates immunity, but there's not actually any pathogen present at all. Now, we haven't had any whole cell viruses in the US since 2008. The last ones were for cholera and plague vaccines. But what are the difference between dead organisms and live organisms? We've sort of covered that already, but dead organisms are the safest because they're dead, and we definitely know they're not going to multiply and produce a further infection that might lead to disease. But they're generally less effective immunogens because they're not alive. And often we're using the wrong portal of entry, injectable, for example. We've already covered that most invaders can't enter through the skin anyway. In live organisms, we do see a better response because they mimic the actual infection, but they multiply and produce infection. Ideally, they do not produce disease. However, this is one of the risky points. They could produce disease. Or in live organisms, there could be other components that unintentionally cause disease, as was seen in the earlier days of the polio vaccine. These live organisms, if they're effective though, tend to confer very long-term protection. Another hazard is they could mutate back to a virulent form, which has been an issue in some other vaccines. Again, this is why in the US we're not using whole cell vaccines anymore. These following couple of tables will give you some insight into the sorts of vaccines that are available in the US. Here they've been broken down into their various categories: subunit vaccines, conjugated subunit vaccines, inactivated vaccines, attenuated vaccines, and then whole bacteria vaccines that we don't really see anymore. Just an interesting list to look at. Nothing you need to commit to memory, but I think this column with the type and deciding whether they're subunit or inactivated or attenuated, at least looking at these can help you sort out that terminology. This next table helps us look at all these multiple disease vaccines, like the DTaP vaccine that we hear so much about. Another interesting table to look at, but again, nothing to commit to memory. Let's have a quick concept check. If you received an antivenom for black widow spider bite, what sort of immunity have you been given? Is it artificial active, natural active, artificial passive? Oops, we're going to have to change that one. Or natural passive. So this will help us think about each of these different categories of immunity. Artificial active. Anytime it's artificial, it's given as a medical treatment. Natural is something that we are assuming naturally without medical intervention. So we can take both naturals off the list. We definitely know artificial active involves activating our own immune response to produce antibodies to whatever particles injected, versus passive, we're injecting the antibody. So here, in this case, I think artificial passive is the best choice. Artificial active would be a vaccine, whereas artificial passive would be immunotherapy, just like an antivenom for a black widow spider bite. So we have indeed been very successful at developing vaccines. But even though we have so many vaccines, most of them are out of reach of much of the world's population, which is a pretty sad thing. In addition to that, there are many bacterial, viral, protozoan, and fungal diseases that remain without vaccines. It's fairly difficult to develop new vaccines for latent or persistent viral infections like the herpes virus. In these types of diseases, the host's natural immunity cannot clear the infection. So artificial immunity has to even outperform the host's response to a natural infection. So that's quite a big deal. It's difficult to choose vaccine antigens that are safe and that properly stimulate immunity. Vaccines take years and years and years to develop and become approved. And even after the approval process, there are still side effects that can come about. For example, there's some really controversial things about vaccination right now. When we look back at the polio vaccine, one of my colleagues is working on some research that shows that there's a very strong correlation between the polio vaccine and mesothelioma. Very, very controversial work, though, because the government required that vaccination. And then the pharmaceutical companies that developed that vaccination. So who's at fault? When you think about vaccines and their necessity, there's definitely some good sides. However, there's definitely some arguments. Now, one of the arguments that's been presented was that there was a connection between vaccination and autism. Now, this has been pretty clearly shown to not necessarily be the case. There could be some correlation which doesn't indicate causation. However, there are definitely some serious implications to vaccination processes. Overall, though, probably generalized vaccination has more positives than negatives. Research these days is focused on newer strategies for vaccine preparation, using things like antigen synthesis, so synthetic epitopes being made, or recombinant DNA, and gene cloning technology. One of the chapters that we skipped contains a lot of information on these types of technologies. Feel free to go back to that chapter and delve into this topic if it interests you. You'll discover a lot about genetic engineering. Genetically engineered vaccines use recombinant DNA technology. That is, we take a gene from a pathogenic microbe that leads to the display of its antigen, and we can insert that gene into another vector in order to deliver it into the host and have that antigen produced. There are things like Trojan horse vaccines, where genetic material from an infectious agent is inserted to a live carrier microbe that's non-pathogenic. Like the Greek soldiers were hidden in the large wooden horse that was used to sneak them into the enemy's fortress. In the same way, an immunization gene is hidden inside this live carrier microbe. The recombinant microbe will then multiply and express the foreign genes, and the vaccine recipient will be immunized against the microbial antigens. So Trojan horse simply means hiding the genetic material inside another microbe. There's a lot of work going on at the moment in DNA vaccines, where foreign DNA is inserted into a host using recombinant DNA technology. Here we have the DNA of a pathogen that codes for one of the antigens, and then it's inserted into a plasmid. This plasmid is then put into an injection and injected into the subject. Once that plasmid is accepted, the DNA is transcribed and translated into these various proteins. Then this protein is expressed on the surface of the cell so that it looks like the pathogen, and that stimulates an immune response, thus bringing around immunity. Let's take a look at a quick vaccine that'll help us understand how some vaccines are constructed.
There are a variety of ways to produce vaccines. Traditionally, vaccines have consisted of killed or inactivated pathogens. A second traditional approach is to use an attenuated strain of the pathogen. During attenuation, the pathogen loses its virulence, but retains many of its antigens and is therefore able to elicit an immune response. Subunit or acellular vaccines consist of selected components from the pathogen, such as a surface structure, an internal macromolecule, or the flagellum. Recombinant vaccines are produced by cloning the gene for a structure, such as a surface protein from a pathogen, into a host microorganism. For example, the gene for a surface protein of the hepatitis B virus has been cloned into a yeast cell. The yeast produces the virus surface protein, which can be purified from the growth medium of the yeast and used as a vaccine.
Vaccines are administered in a number of different ways. Most of them are injected by subcutaneous, intramuscular, or intradermal routes. Only a very few vaccines are available in oral form, even though they have very strong advantages, particularly that they're entering through the correct portal of entry. Some vaccines require addition of other substances, like an adjuvant, which is a compound that binds the vaccine and improves its immunogenicity. It can hold the antigen in the tissue for gradual release. For example, we might use an emulsion of mineral oil, water, and then some mycobacterial extracts or beeswax to hold it into the tissue so that it can be released gradually. As I've already shed a little bit of light on, there are many side effects possible from vaccines. Some of the simpler ones include local reactions at the injection site, fever, and allergies. However, at the time of release, we really don't know the long-term side effects of many of these antigens. If you want a really good read, check out a book called The Virus and the Vaccine, a true story of a cancer-causing monkey virus, contaminated polio vaccine. It's written by Debbie Bookchin and Jim Schumacher. Fascinating book about the history of that polio vaccine. Involves discussion of much of the research of my friend Dr. Michele Carbone. Again, lots of controversies around vaccinations, but the benefit probably really outweighs some of these side effects in the population in general. So why should we vaccinate? Who should we vaccinate? And when should we vaccinate? The table on the right gives you some idea of the periods of time in which people should receive vaccinations. These are the ones that are recommended by the FDA. Vaccination not only confers protection to the individual receiving the vaccine, but it also protects public health. And the key here is the idea of herd immunity. If we get the majority of our population immunized, people who choose not to immunize can also be fairly safe. This collective immunity through mass immunization confers indirect protection on the non-immune members. If most people are immunized, one person that's not vaccinated may get the disease. However, it can't be passed on to others because they've been immunized. And there are figures, depending on the pathogen, of how many of the population need to be immunized to get this herd immunity. This is a really important force in preventing epidemics that doesn't exist in some of our third world countries. So if you are traveling, it's extra important to get these immunizations because there isn't herd immunity to prevent it being passed to you. People that have it can pass it to you easily and you can bring it home, as you'll see in the case study in this chapter. Anyway, that brings us to the end of this chapter. Lots of really fascinating information, difficult stuff to get. So please spend as much time as you can in Learn Smart and using the other tools within Connect, within the chapter of our text, to get a grip on this material. I highly recommend diagramming or mind mapping to get a good understanding of this material.